Passive agglutination, the CRP Latex Agglutination test. The CRP test begins with the Immunex CRP latex reagent, a negative control, a positive control, and a patient sample. You will also need a clean black ceramic slide that is supplied with the test reagents. In this lab, the slides being used have 6 circles. We will only be using 3 of the 6 circles. In the 3 circles you will be using for your tests, you will place 1 to 2 drops of the latex CRP reagent. It is important to add the reagent to the slide first to prevent contamination with any of the controls. It is also suggested that you place your controls in the 2 outside circles and your patient sample in the middle. This will make observation of your results easier later on. Notice with the addition of controls that the tip of the dropper never touches the reagent in the circle. Only 1 drop of control is needed. Now being added is the patient sample in the middle. Again, the tip of the dropper never touches the reagent on the slide. Using a clean toothpick, mix the reagent and the sample on each of the circles. Do not cross-contaminate the circles with each other. Notice that the controls are being mixed first. A new toothpick will be used for the patient sample. Once the reagents and controls and patient sample have been thoroughly mixed, you will need to roll the slide back and forth. This is done by gripping the slide in both hands and rotating it in a clockwise or counterclockwise manner. This should be done for 30 seconds to 1 minute. Shown here is a diagram of an antibody-antigen reaction. In an agglutination reaction, or passive agglutination reaction, antibodies have been attached by their stem region to a small microscopic latex bead. As the beads covered in antibodies are rolled around and mixed with the antigen solution, an agglutination reaction will occur. One antibody may bind more than one antigen. This causes a clumping to occur in the solution. Clumping is visible in the CRP Latex Agglutination test. Shown here are the reactions from the CRP Latex Agglutination test. On the left is a negative control, on the right is a positive control, and in the center is the patient sample. By placing the patient sample in the middle of the 2 controls, determining results of your patient sample is much easier. This is a picture of the positive control of the CRP Latex Agglutination test. The small white clumps seen are due to the agglutination of the latex beads with the antigen present in the positive control. Now shown is the reaction in the negative control. The solution remains a milky white and no agglutination is visible. Here we compare the positive control to the patient sample. Would you consider the patient positive or negative for the presence of CRP? The Immunex CRP test is a form of passive agglutination used to detect the presence of C-reactive protein in patient serum. C-reactive protein, also known as complement reactive protein, is only produced by the body in response to bacterial infections or rheumatoid arthritis. The Immunex CRP Latex Agglutination test is used as a confirmatory test for rheumatoid arthritis after all bacterial infections have been ruled out.